Hi everyone, my name is Emma Tobin and I'll be presenting Figure 5. I wanted to provide a recap before we actually get into Figure 5 because I know that a lot of data and a lot of results have been reviewed so far. So first, in Figure 1, the results indicated that Cas9 is a DNA endonuclease, and that is guided by two RNAs called tracer RNA and CR-RNAs. In Figure 2, we saw that each Cas9 nuclease domain cleaves one DNA strand. In Figure 3, we saw that there are various requirements for the tracer RNA and the CR-RNA to guide the Cas9 for DNA cleavage. These involve conservation of native nucleotide sequences at certain lengths and positions. And the protospacer adjacent motif was also found to be an important structure in cleavage activity. In Figure 4, the PAM motif was further investigated, and it was found that PAM was only needed in cleavage of double-stranded and not single-stranded DNA. This hinted towards possible involvement in strand separation, strand invasion, or R-loop formation. The authors wanted to know how their findings could be used for genome editing and programmed DNA cleavage. The authors therefore hypothesized that the dual RNA-guided Cas9 system could be simplified by making a chimeric RNA that could join to the ends of the tracer RNA and the CR-RNA. They designed one long chimeric RNA and one short one, which retained the target recognition sequence and incorporated a hairpin structure for the base pairing between what would be the CR-RNA and the tracer RNA. And we can see this in Figure 5a, in which the top picture shows the naturally occurring system. We can see tracer RNA here and CR-RNA here. And in the engineered model that they showed, you can see that there is one long strand of chimeric RNA, and it has similar features as the cRNA and the tracer RNA, but it also incorporates this linker loop, which makes that hairpin structure. So first, I want to talk about the methods that were used to obtain the data before I actually get into the results. Some other people might have already talked about plasmid DNA cleavage, but we can review it briefly here again. So the first step in plasmid DNA cleavage is the annealing of CR-RNA and tracer RNA. And additionally, a plasmid must be linearized first, or at least it was in the case of these results. I couldn't find any figures or pictures representing this method online, so I made my own, and I hope it gets the point across. You can see here that there's a circular double-stranded DNA plasmid, and there is a restriction enzyme represented in green. And the restriction enzyme will cut the plasmid at a specific site, and it will linearize it. This linearized plasmid can then be incubated with Cas9 in the annealed complex. And Cas9 will then cut the linearized plasmid, and this cut will result in two pieces of double-stranded DNA, one longer and one shorter. And due to the size difference, they can be separated using agarose gel electrophoresis. I'm sure many of you have become familiar with gel electrophoresis over the years, but I want to go over it again just to make sure everyone has an understanding of this procedure. If you were making your own agarose gel, the first thing that you would do is pour agarose solution into the electrophoresis setup. And you would then use a comb to create wells into the agarose gel. You would allow the agarose gel to cool and thereby solidify. And you could then pipette your samples into their respective wells. Once you turn on the power, the DNA fragments will travel from the cathode to the anode. And this is due to the fact that DNA has a negatively charged phosphate backbone. And different bands will appear in different lanes because various sizes of DNA fragments will be present. As we saw on the last slide, the Cas9 enzyme cut the DNA into two different sizes of fragments. So these two fragments would travel at different speeds. The smaller fragment will travel faster and the larger fragment will travel slower. Now we'll actually be getting into the results of Figure 5. And we can see on the right-hand side that Figure 5b is displayed. we can see in the top portion that the separation by gel electrophoresis has been visualized and imaged using ethidium bromide, and that the designs of Chimera A and Chimera B are displayed in the lower portion. We can see the differences between the two in the blue part, where Chimera A is longer and Chimera B is shorter. The linearized plasmids without cleavage by Cas9 was about 2,640 base pairs in length. And once they were cut, they were approximately 1,880 base pairs and 800 base pairs. We can see in the leftmost lane that there was a ladder that was used to determine how many base pairs were present. And in lane 2, we can see that CRRNA and tracer RNA are not present, but Cas9 is present. And the full-length linearized plasmid is there, this band that is there, and it shows that Cas9 did not cleave the linearized plasmid. And this was also shown in earlier experiments, but this is used as a control for this gel. In lane 3, we can see that the photospacer 4 version of CRRNA and the 4 to 89 truncated version of tracer RNA alongside Cas was used. And unlike lane 2, we can see that there are bands present at 1,880 base pairs and 800 base pairs, which shows that Cas9 was effective and was able to cleave the linearized plasmid into two pieces. Now looking at lane 4 and lane 5, we can see that Chimera A, the longer Chimera, was very similar to lane 3 in that bands appeared at 1,880 and 800 base pairs, which shows that Chimera A was able to cleave the linearized plasmid. On the other hand, Chimera B is more similar to lane 2 than it is to lane 3, and a band only appeared at 2,640 base pairs, which means that the plasmid was not cleaved. And therefore, Chimera A was effective at cleaving, and Chimera B was ineffective at cleaving. I'm going to be talking about the methods again because the methods for the next portion of Figure 5 differ slightly than the methods that were used for Figure 5b. So this cleavage assay that was used was very similar to the plasmid cleavage assay, but instead of a plasmid, the researchers used a short synthetically made DNA oligonucleotide that was radioactively labeled on the 5' end of one strand. And once again, I could not find any figures or pictures for this method, so I made my own. And we can see that this purple triangle is the radioactive label, and it's only on one side of the rectangle that's representing the double-stranded DNA. And an active Cas9 would come in and cleave at a certain position, which would result in two different double-stranded DNA fragments of various sizes. And one of these would be labeled with the radioactive component. I also noted here that the full-length oligonucleotide is 50 nucleotides in length, and the cut version that was radioactively labeled is 13 nucleotides in length. Now I'll be getting into the results corresponding to that method. And we can see on the right that Figure 5c is displayed. Figure 5c supports the results seen in Figure 5b, and I will go over that in more detail. We can see the topmost bands are 50 nucleotides, and the bottommost bands are 13 nucleotides, which is the uncut and the cut portions with the radioactive label. We can see that lane 1 is a control in which only Cas9 was implemented, and the oligonucleotide was not cut because the band is at 50 nucleotides. Lanes 2 and 3 show a full-length CRRNA for protospacer 4 and a tracer RNA that was truncated. And Figure 3 shows that these two truncated versions of the tracer RNA work and are valid uses of the control. And we can also see this here too, where the oligonucleotide is cut and the band appears at 13 nucleotides. In lane 4, we can see Chimera A was implemented, and Chimera A was able to cut the oligonucleotide because the band is at 13 nucleotides. And Chimera B was barely able to cut the oligonucleotide. I think there's a faint band, but for the most part, it was ineffective at cutting. And because the cut bands were at 13 nucleotides, at least for Chimera A, we can conclude that the cleavage site is the same. If it wasn't the same, then the resulting product would be a different length and would appear at a different area of the gel. Once the researchers knew that the chimeric RNA was effective at cleaving oligonucleotides and plasmids that had protospacers in conjunction with Cas9, they wondered whether the chimeric RNAs could be reprogrammed to target genes of interest. And in this case, they chose to target the green fluorescent protein gene, which they implemented inside a linearized plasmid. And the plasmid was linearized using similar methods as seen in Figure 5b. At its full length, the linearized plasmid was 11,100 base pairs, and it was 7,400 and 3,700 base pairs when it was cut. The authors designed 5 different chimeric RNAs, which can be seen in Figure 5d on the right. In the leftmost lane, we can see a ladder to indicate how many base pairs corresponded to each signal. And the 5 lanes after that showed the different chimeric RNAs that were implemented alongside Cas9. And if we compare it to the rightmost lane, in which no chimeric RNA was present, and therefore the linearized plasmid was kept at full length, there are 2 bands present for each chimeric RNA. And that means that the chimeric RNA was successful at working with Cas9 and cleaving the plasmid at a specific target site. So there are a few conclusions that the authors were able to draw from all these experiments. I've already talked about most of these, but I'll highlight them here again. So first, as seen in Figure 5b and Figure 5c, Chimera A was suitable for DNA cleavage, whereas Chimera B was not suitable and was not effective at cleavage. And the difference between these 2 chimeras was their length. So this suggests that when designing chimeric RNAs, one needs to consider the nucleotide length that is used. In Figure 5c, we saw that the cleavage site remains the same between the dual RNA guided system and the chimeric RNA guided system. And lastly, chimeras could be successfully engineered to target a gene of interest. And this was seen in Figure 5d when chimeras were engineered to target the green fluorescent protein gene. Overall, the authors concluded that the system is robust and only has a few constraints, including nucleotide length and the recognition sequence. And overall, the chimeric RNA is a useful tool.